regards to genetics of substance use disorder, an overview. So, I mean, this is the brief outline I'll be doing. So, I mean, based on the previous case, I mean, we would actually understand the importance of a gene environment. Here we had the boy with significant family loading of different psychiatric illnesses and some excessive behaviors that were there. So, I mean, any substance use disorder risk is influenced by both genetic and environmental factors, and there is always a necessary component of gene by environment interaction. So a person cannot become whatever his genetic makeup substance dependent without exposure to the substance. So that is the most important point. So here is the importance of the gene environment interaction. So a lot of the times the risk factors for substance use disorders influence individuals to select themselves into environments that can convey a high risk for substance use and abuse. So this can be illustrated in social construct like peer deviance or this thing, in the sense that I mean they may be a lot of the times in early onset drug dependence, we see a lot of externalizing symptoms, and people who start using drugs actually move in their own circles. So this actually here you can see an interplay between the genes and the environment, where they have a particular genetic or constitutional makeup, they find a particular environment in where the substance use actually increases. So the higher risk environment here would be mediational, sitting in the causal pathway between genes and an end phenotype of substance use disorder. So basically there is two pathways inside the skin. So based upon the genetic makeup, there are different mechanisms by molecular biology, or outside the skin, where the genes, because of the genes, they go to a particular environment and personality traits where again, once they are in that environment, it actually increases the risk of progressing to substance use disorder. So a lot of the time, environmental conditions that affect access to substance use moderate the degree to which genetic influence acts. So there are a lot of countries which, some countries like Nigeria, which actually, regulate the use of tobacco and all. So here nicotine dependence in younger people is not as much as what is found. So the environment plays a large role in actually determining the risk of a substance use disorder. So environments that are more controlling provide greater social structure and reduce the impact of genetic risk of a substance use disorder. There is something called a social expression or social triggering, where some environments allow greater opportunity to express genetic predispositions. And also there is a lot of time a connection to stress, which is another factor which may trigger substance use. So I'll just go to the next slide. So, I mean, the genetic factors so in two ways, genetic factors can play in two ways. They may actually moderate a role in initiation of any substance use. Also, once the substance use is initiated, there might be a switching on of some genes, which would actually lead on to a progression and to a higher degree of substance use disorder that is there. So this would be explained in the coming sessions, and epigenetic mechanisms and all. So generally what is found is that shared environmental factors generally affect the initiation of any substance use, but not progression. So the dependence, the use in adulthood, these factors are actually related to a greater degree to the genetic factors that are there. Also, the other thing is there is significant overlap in genetic influences between different substances, and also the liability, genetic liability to the so-called externalizing disorders is something else, apart from the independent risk factors for a substance use disorder, which actually leads to the genetic makeup. So I'll be just going on to the various levels of studies. So initially it was with regards to family studies. So numerous studies have found that the rate of alcoholism are substantially higher in relatives of people with alcohol use disorders. But however, the drawbacks of family studies are they can also reflect the shared social and developmental influences. So hence these are inconclusive. Now twin studies would actually, there would be three, so it would show the genetic effects that are there among the twins, the common environmental effects that are shared by the twins, and specific non-shared environmental exposures. So results in twin studies are varying, where 33 to 71% of nicotine dependence may share a genetic vulnerability, the variations, and 40 to 66% in alcohol. So the concordance rates in monozygotic twins are higher than in dizygotic twins as well. So these are the three. I mean, in genetic, distinct twin studies, it may be because of the genetic risk that is there, it may be because of the shared environment or unique environment, where a specific one of the twins is exposed to a particular environment which has a higher risk of substance use, as discussed earlier. So the variations, there is a large degree of variations that are there in the heritability that are there. This may be because of the stages of addiction. So addiction is a multi-stage process. So as earlier discussed, it's the environment which has a large part in initiation, but actually progression and actually into a dependent syndrome, it actually is based on genetic factors. So for measures of quantity frequency, the heritable factors seem to play a greater role. And when these actually develop in adulthood, it would actually explain a higher degree, about 75% of variation among individual differences. So this is that. So there is another method would be adoption studies, where subjects who had a biological parent with severe alcohol problems were significantly more likely to have alcoholism than if their surrogate parents were alcoholics. So here in the adoption studies, the rationale would be that the biology, that is, they would be born to a parent with a substance use disorder, but be in an environment where the surrogate parents do not use substance use disorders. And this is compared with a parent who did not have substance use disorder, where the surrogate parents had a substance use disorders, and the former was found to have a higher degree of substance use. So this is another mechanism. Then later on, linkage studies. However, this is of more of historical interest. We know most of the psychiatric disorders have a common disease, common variant model. Linkage studies is for more rare disorders, where genetic mutation will affect the phenotype 5 to 10-fold. So, I mean, though this is of more of historical interest, linkage studies are more of historical interest, and it's not a good method for the kind of common disease, common variant model which we propose for most of the psychiatric disorders. So a lot of the time, what obscures in identifying a different phenotype by genes, or what getting clear, or why do we get heterogeneous results? So a lot of the time, there is a lot of clinical and etiologic heterogeneity. So as discussed earlier, there are a lot of environmental factors, as discussed earlier, based on the age. As adults, in adults, the genetic influences, for adults who are substance dependent, the genetic influence actually explains about 75% of the variance. In contrast to someone who is a much more younger substance user, where the shared environmental factors also play an important role. So in most of the studies, significant and the diagnosis we use is the number of days of abstinence, or how many. So these are weak and inconsistent. And they would require very high sample sizes to achieve any kinds of significance. And so this has led to this thing where we are looking at for dimensional ways, the way dimensional any psychiatric disorders can be described dimensionally. So in dimensions, even with people without substance use disorder, a value can be ascertained, and so it would be in a spectrum, and this would help us to better identify particular candidate genes. So different types of phenotypes that are used are based on types of alcoholism that might be there, Cloninger subtypes, Babor subtypes. There are a lot of endophenotypes. Endophenotypes are markers between the genotype and the phenotype. So these are intermediate markers, and these were described by Gottesman and colleagues. So these are heritable, they co-segregate, and also they co-segregate, and that's state independent. So they would be there to some degree even in individuals in the family who did not have the particular phenotype. So one well-studied marker is amplitude of P300, which is reduced in substance use disorder. Also other dimensions in addiction, like tolerance, cue reactivity, a lot of neuropsychological indices are actually used as endophenotypes to actually study better the genetics. Level of response to alcohol is another phenotype. This was studied well by Shakit and colleagues. That is another phenotype. And imaging genetics. So patterns of brain activation in fMRI studies or PET studies. And it's basically use of neuroimaging to characterize gene effects. So the neuropsychological tasks like delay discounting, and where you see that which is a delay discounting is one of the domains of impulsivity. So it is associated with dopamine signaling, and several functional polymorphisms have been identified in the dopamine transporter gene and the dopamine receptor D2, which influences the activity in ventral striatum. So this is also help in making varied phenotypes. The next level of study is candidate gene studies. Candidate genes, basically, they allow allow researchers to investigate the value of an educated guess about the genetic basis of a disorder. So, and basically here, the thing is that we should know something about the biology. So in candidate gene studies, for example, one thing initially what in Alzheimer's disease, where in the neurofibrillary tangles, ApoE was there. So, since it was there, it was studied. Or in alcohol, since in alcohol metabolism, alcohol dehydrogenase and aldehyde dehydrogenase, these are specific genes that have a role in the biology, and that can be studied whether it actually confers any kind of genetic vulnerability, whether any kind of polymorphisms, mutation. So this is how candidate gene studies are done. So the advantages would be do not require large families, both affected and unaffected members, which would be there in linkage studies. The different steps of selecting a candidate gene, you would have to choose a DNA polymorphism. What is the specific SNP? What are the different variants of the SNP in the population? That needs to be known. And then testing the particular candidate gene. That would be the different steps. So in alcohol, with respect to alcohol and aldehyde dehydrogenase, the alcohol flush reaction. So these are some of the things that had come on through different candidate gene studies. And it is also described to some degree in cannabis and OPRM1 in opioids. Now, Genome Wide Association studies differ from candidate gene studies in that there is no a priori hypothesis. So it's an unbiased assessment of the entire genome. So the entire genome based upon the, so initially there was the HapMap project, that's the haplotype mapping, where the number of SNPs, so more than 2.5 million SNPs were actually coded and detected, and the different variations in different populations were actually noted, and whether it was in any kind of linkage disequilibrium with genes that may be of interest. So when you actually studied in persons with risk, you would actually come across some SNPs of interest, and then you would actually look at regions associated with these SNPs to find out whether there are any genes which actually may relate to two, and then you'll have to actually study the effects of these genes. So this is how genome-wide association. The disadvantage, however, is since there are multiple markers, so around 2.5 million SNPs, to reach any level of significance, you would require a very low p-value, where this thing where in the region of 5 into 10 to the power 8, because of the large genetic markers that are tested to reach any level of significance. So GWAS, however, has held, especially with related to, it's well described in smoking and all, alpha 5 subunit nicotinic acetylcholine receptor, it has helped, and there have been a lot of meta-analysis of different studies to actually identify. So this is one well-known meta-analysis where the Tobacco Genetics Consortium and the group by Liu et al., they combined their data, and there was a lot of this thing about the nicotinic acetylcholinergic receptor, where alpha 5, alpha 3, and beta 4 locuses, which were there in 15. So this is some of the contributions of GWAS. With relation to alcohol, something with related to autism susceptibility candidate 2 gene, some importance was seen. So, but however, even with GWAS, the thing is that it can only explain about 2% of the variance that is there. So, the thing is that since it actually overlooks the evidence for polygenic association. So there will be multiple genes. So some of the genes, like there are a lot of times cell addition, genes for cell addition and all are implicated because they actually affect the functioning of various genes and proteins. These actually may not actually reach levels of significance, but however may be involved. So that is a drawback. We'll just skip the slide. So basically, GWAS has 2 points, but there are it does not represent rare SNPs like in alcohol dehydrogenase. So some of these rare SNPs associated with these genes are not represented in a lot of the GWAS. The subsets that are there. So we may be missing out to some degree. So I would just, so this would, so GWAS mainly looks at single nucleotide polymorphism, but also there are something called as copy number variants, which are larger segments of DNA which are deleted or duplicated, which has considerable structural integrity and which needs to be explored. And also, as mentioned earlier, rare variants are inadequately captured on commercial GWAS arrays. So also epigenetic modifications are also implicated. So this may be why we are not able to explain all the variants that are there in genetic through GWAS studies. Just these slides. Then the other important thing is pharmacogenomics, which would be dealt in detail in the last session. So some of the well-described this thing is with relation between OPRM1 and naltrexone. So that is something that is very well described. And then there's GATA4 gene polymorphisms associated predicting treatment response to acamprosate. And Greek 1, which was associated with severity of topiramate-induced side effects. So there are a lot of pharmacogenomics will be there, and will be discussed in detail in the final session. And finally, related to epigenetic changes. So basically, it can affect in 3 ways. So repeated exposure of a drug abuse in adolescence causes addiction in vulnerable individuals by inducing stable changes in gene expression. So it can cause, when the exposure to drugs is in adolescence, it can cause stable changes in gene expression. Or epigenetic regulation can induce certain genes and it can change the steady state gene expression the other way around. Or because exposure to drugs during pregnancy or this thing, these induced epigenetic changes might be transferred through sperm or ova. So these are the 3 ways different epigenetic mechanisms can act. So it acts to post-translational covalent modifications of histones, or DNA methylation is another thing that is there. So the multiple and varied epigenetic mechanisms which act. So this is just a picture. So the nucleosome is where the 147 base pairs of the DNA are bound around the histone octamer. So according to the DNA methylation acetylation, these actually wind or unwind. So generally acetylation leads to the histone acetylation leads to unwinding of the DNA, and actually the mRNA is actually formed because of that. A lot of the time methylation actually represses gene expression, and based upon it, it is related to a lot of other genes downstream, like the C response element binding protein, which reduces the sensitivity to drug with related to Q-induced stressor, or the delta phosph, which increased expression increases the sensitivity to and responses to drug exposure. So it affects multiple and major molecules downstream, which are very important in development of addiction. Yeah, so these are my references. So, there has been just some degree of time, and what I've tried to do in this particular presentation are try to the different levels at which genetic studies are done. There are a lot of things that I have not covered, but in the coming sessions, which deals on genes and then phenotyping and pharmacogenomics, they will be dealt in. So these are some of my references which are there. Yeah, thank you.